Chemical tests of meat, lecture 2. In this lecture, we will learn how to determine pH of meat. The pH of a meat product can reveal a great deal about the product. To understand how, it is important to understand how muscles become meat. When an animal dies, the muscles the muscle will continue to contract and relax. This is often seen immediately after exsanguination as twitching muscle. The carcass at this point is dying muscle. This process of contracting and relaxing requires energy which is achieved from glycogen that is stored in the muscle. In living muscle, the muscle glycogen stores are continuously replenished as they are used. As blood is no longer circulating in the exsanguished animal, the muscle glycogen stores become depleted with the time. As they become depleted, the muscles stay in a contracted state as there is no longer energy available to relax. This is rigor and is commonly seen in recently killed animals. With the time, muscle fibers began to break down and the muscle tenderizes and become meat. Because there is no circulating blood, there is also no circulating oxygen in the dying muscle. When the glycogen is used by the muscle in an anaerobic environment, instead of glucose, lactic acid is produced. As more lactic acid is produced, the pH of the dying muscle decreases. The pH of living muscle is around 7 to 7.2. The pH of dead muscle is ideally from between around 5.6. The pH of meat has a wide range of effect on the meat. When the pH drops too quickly or too slowly, there can be significant changes in the color, water retention, tenderness, and shelf life of that meat. It is for this reason that animals are normally rested after transportation in lairage. The transportation process is stressful and animal will deplete much of their glycogen stores. When the animals are immediately slaughtered upon arriving at the slaughterhouse, the remaining glycogen levels are rapidly depleted. Little lactic acid is produced and the pH does not drop adequately. Resting the animals for 2 to 3 hours will normally allow the muscles to restore their glycogen, allowing a gradual drop in pH and improved meat quality. If the animals are stressed or overly excited just prior to exsanguination, the pH can drop too quickly or too slowly. This is prevented by keeping the animals calm prior to exsanguination. The biggest impact of pH of meat is on water retention. The majority of the water in the meat is bounded loosely to the proteins. This is due to the dipolar nature of the water, allowing them to interact with any charge of the protein. Low pH. When pH drops too low and too quickly, protein can become denatured, losing their function and ability to bind to water. In addition, as the pH lowers, the protein loses their charges. Here, again, the protein lose their ability to bind to water. And there can be high water losses. When the pH is so low, below 5.4, that the meat starts to lose water. It will have a wet appearance. Additionally, the package will also become wet. Meat itself may be pale in color due to loss of myoglobin with water and is often soft. This type of meat is often called PSE, pale, soft and exudative. The main causes of PSE meat are genetics, animals that are excited or stressed right before exsanguination, which cause a very rapid depletion of glycogen. This problem can be increased if the carcass are not refrigerated after exsanguination. If pH becomes too high, in this case, a high pH, which is above 6.5, can significantly increase the water holding capacity of meat. This occurs as more protein retain their charge and tightly binds the water. When the final pH of meat is too high, the meat can appear dry as large amount of water are bounded within the muscles. Because the meat is retaining too much water, it can also appear very firm. Meat with high final pH can often appear dark red in color. This meat is often referred as DRD, dark, red and dry. Measuring pH of meat sample. It's a standard practice. A designated pH meter for meat can be used for this purpose. pH meter where the electrode can be inserted directly into the meat tissues. pH can also easily be measured using a standard pH meter using the same protocol. For this, we need a meat sample, pH meter, blender, and distilled water. First, we need to calibrate the pH meter. Process of calibration has already been practiced in previous practicals. pH meter need to be calibrated routinely. A proper calibration prior to starting an experiment can save a lot of time and produce more accurate results. Different pH meters have different protocols for calibration. Calibration should follow the manufacturer's instruction. So, for measuring pH of the meat sample, first cut the meat samples into small pieces and measure about 10 grams. Combine the meat sample with 100 ml of distilled water and then blend it at a high speed for about 30 seconds to form a uniform slurry. Transfer the meat slurry to beaker and add a magnetic stirrer bar. Then measure pH with the sample while the sample is mixing. While doing this, you should be careful with the pH electrode as the stirring magnet is continuously moving within the same beaker. So avoid hitting that pH electrode with that magnet. Thank you so much.